Welcome to Penrose Learning, where we make understanding easy.
In this video, I will explain how to process embryonic cells. First, ensure all equipment is sterile to prevent contamination. Next, carefully collect the embryonic tissue using fine instruments, minimizing any mechanical stress. Immediately transfer the collected cells into a suitable buffer solution to maintain their viability and prevent degradation. This initial step is crucial for preserving the integrity of these delicate samples for subsequent analysis. We'll cover these steps in more detail. After careful collection, transfer the embryonic cells into a cold buffer solution, ideally at 4 degrees Celsius, to slow down metabolic processes. If your research requires individual cells, gently dissociate the tissue using enzymatic or mechanical methods. This step is critical for obtaining a uniform cell suspension, which is essential for many downstream applications. Always work quickly to minimize the time cells spend outside their optimal environment. Once collected, cells need fixation to preserve their structure. A common method involves using a 4% paraformaldehyde solution for about 15 to 30 minutes. Following fixation, cells often require permeabilization. This process uses detergents, such as Triton X100, to create small pores in the cell membrane. These pores allow larger molecules, like antibodies, to enter the cell and bind to their specific targets for subsequent staining. The final steps involve staining and analysis. Staining uses specific dyes or fluorescently labeled antibodies to highlight cellular components, such as DNA or proteins. Incubate the cells with primary antibodies, then with secondary antibodies conjugated to a fluorophore. After washing, mount the stained cells onto microscope slides. Use a fluorescence microscope to capture images, allowing for detailed observation and quantitative analysis of the embryonic cell structures.
Thanks for watching. If this was helpful, leave a like and consider subscribing. See you next time.